Welcome back to the Podovirus podcast. This is Jessica. This is the third episode. I'm really excited that you're back listening. This interview that I'm going to share with you today is with Jesus Fernandez of Eligo Biosciences. They're a biotech company in Paris, France. I've been following them for several years now. They've been working on using modified phages to target bacteria, especially with microbiome in mind. And they've just published a new paper in Nature on in situ targeted base editing of bacteria in the mouse gut. So this came out July 10th of this year. And so what they show in this paper is that they can actually modify E. coli and Klebsiella in the mouse gut, and they do base editing, which is different from CRISPR. So with base editing, you are just modifying one nucleotide at a time without cutting the DNA. You're just editing it out. And so, as we'll find out, that's crucial for microbiome editing because with CRISPR, you're going to kill the bacteria when you try to edit. And so we talk about the history of Eligo, what they're up to, how they think about what their technology, now that they've kind of gotten it working so well, what it could be used for, and some of the different things like cancer, Alzheimer's. It's kind of a shift in thinking from phage therapy, which I think about a lot, to actually using phage-based technologies to fix other diseases. So things that have nothing infectious necessarily, they're not considered an infectious disease, but they involve a bacterium or a bacterial gene that is made in the body that has some impact. And that can lead to some disease. So it was really great to talk to Jesus and just probe him with all my questions about where we're at with the microbiome, with editing genes, the different technologies. Like it's not just CRISPR anymore, and all the ways that we could be sort of using what we know about phages to have impacts on a lot of different diseases. With that, here is the conversation with Jesus. All right. So, hi, welcome. I feel like Eligo has been just a company that I've been having my eye on ever since we started Phage Directory in 2017. I don't remember when exactly started, but it was in the early days of me getting into this space. And Xavier, he was always kind of at the Phage conferences. And I just always been rooting for you guys and meeting your scientists. And it seemed like you had a really cool mission. And all of us in the phage space, I feel, are hoping for successes so the whole field can be lifted, and definitely looking at using phages to edit the microbiome. I don't know. I guess I want to know if that was always your focus or was it more antimicrobial, but also like how it led to where you are now.
Yeah, no, so you're very right. So, Eligo, so I started Eligo almost 8 years ago, or more than 8 years ago now. Eligo started as an antimicrobial company. So it was, David Vicar, one of the co-founders of Eligo, that was working with Xavier, and then he got this paper out where he could like use CRISPR delivered through a phage that would kill bacteria through a sequence-specific killing, right? So that was the start of Eligo. So um, we had a lot of success doing that. Actually, had Eligo using modified phages to deliver a payload that contained CRISPR. So it's important to know that what we do at Eligo is not a phage. It's not really directly a phage. So it's not. So it's based on the classical cosmid technology, uh, 2.0, let's say. Okay, so we apply all the synthetic biology techniques that we know today to build particles that are really good at delivering and with circuits that have been engineered to do exactly what we want to do. So we started using CRISPR, sequence-specific antimicrobials. We had several programs on that. And then we realized, with all the CRISPR technology booming in the last years, we realized that actually our technology was great to do not only that, but a way of modifying the microbiome, not only with having specific killing in mind without disturbing the rest of the bacteria that live for example, in the gut or in the skin, but also to make them express things. So we can modify the phage meats and the Eligo biotics actually to contain payloads that are going to be expressed by the target bacteria, or in the case that this is what we show in the paper, to modify genes in situ without the need of making really large disruptions in microbiome, like no antibiotic treatment, no replacement, so directly in the complex uh, environment of the natural microbiome. And so that was a little bit the history of Eligo, how we moved to one thing to the other, and we have several programs that really touch upon these three aspects of the technology, which is base editing or something in situ, killing bacteria, sequence specific killing of bacteria in the native environments, and modifying the bacteria so they express something also in the native environments.
Okay, awesome. So you edit and then you kill or you modify, if you could think of it like that. Okay. I guess what I want to dive into, and I've always wondered this about anything to do with microbiome therapeutics, if you call them that, is to what extent is it known for all the things that ail us, diseases across the board? How much is it a bottleneck deciding what to target versus targeting it?
Yeah. So that's a really good question. So, and that's actually the difference between. So whenever I talk about what we do at Eligo, people, CRISPR killing or editing, base editing, people really have human genes in mind. So there's a human gene. We know a lot about these mutations that cause diseases in humans. And the thing is that this is kind of a paradigm shift. It's not just having a human with a patient with a mutation in a gene that you can edit. Um, we have realized, like the last years, that also the genes that exist in the microbiome are very, very important for human diseases. Is exactly what Eli was trying to do: bring this technology to patients by touching upon the genes that exist in the microbiome. So there's not a lot of specific targets known yet for bacterial genes. So we know bacteria cause diseases. Could be degenerative diseases. It could be cancer. They could affect, for example, a treatment. A patient is following like an anti-cancer treatment. Bacteria can modify the molecule and make it inactive. They can sequester it. So they can really do a lot of things that affect the health of patients. But it's hard to pinpoint exactly which bacteria are doing that and what's a gene that is causing that. And I think so. There's a lot of people working on this right now, and I think the technology that we show in the paper provides a really, really great way of interrogating specific gene functions in a natural environment. So, for example, you say, okay, I think, or we have some idea, or somebody, we're collaborating with somebody, and we think this bacteria might have a gene that does X, Y, Z, has an impact in the patient. So we can really go there, modify the gene, inactivate it without killing the bug, and see if that has an effect in the disease, for example. So I think it's a really great way of interrogating, um, having this kind of correlation between a gene and an impact in disease. So I think there's going to be a shift in the way we do things. And I think that's exactly what we need, finding these specific targets.
Yeah, that's so interesting because, yeah, it just seems like, I mean, I'm wondering, has it been not known? Like, do we not know targets because there haven't been the technologies to probe them and to interrogate them? That's maybe what you guys are saying.
Exactly. That's one of the reasons. You can do things in vitro, and you can say, oh, I put this bacterium together with these cells, and something happens. That's never going to be a real disease setup. For example, in an animal model, where you have a disease model, some bacteria cannot be cultivated in vitro. So you might not be able to really do that directly because they need to be part of a consortium, or it's really just not possible to cultivate them. Um, so at the end, having this technology that allows it to do something in the natural environment, to me, is the key to really find specific targets that we can go after.
Yeah. And like touching on what you said about uncultured organisms and just like all the bugs that we barely know anything about because they don't play well in the lab or in animal models. How, yeah, like can you use your technology equivalently on those kind of targets, or is it going to be the same problem? How do you think about that?
Yeah, so that's a good question. So actually, we talk about that in the paper, and while we focus on enterobacteria here, I think one of the most important aspects of what we do is that you don't need to have a specific vehicle, specific elibiotic is how we call it particles. So you can program to target just E. coli. We show you we can jump the species barrier as long as they're relatively close. For example, we jump from E. coli to Klebsiella. We can edit Klebsiella using a lambda-based particle, by changing some of the receptor binding terminants. So yes, different targets will most probably need different vehicles to be developed, different, uh, uh, an understanding of what we need to bind to make them deliver well to what we need, and that's part of the process, But I think what we show here is the first step in that race, and that will lead us to really bring this technology to the clinic, which I think is it would be great to have somebody treated with this kind of technology and cure a disease, right? This is what we are after. So
Yeah, it's exactly. I want to talk about like how do you distinguish for someone who's not in the CRISPR space, gene editing space, is more of a phage biologist. Um, base editing versus CRISPR. Like how do we think about the different technologies and the types of ways you're gonna edit something?
yeah, so that's a good question, and I go back to what I said before about the image that people have about CRISPR on eukaryotic cells or human cells and bacteria. So I think the main difference here is when you express CRISPR in a human cell, CRISPR will Cas9 nuclease, will clip the genome, and the repair mechanism of the cells, the human cells in this case, will repair that, and the cell would not die, will stay alive, and will have a modification. Well, the vast majority of bacteria don't have this kind of repair mechanism. So if you cleave the genome of a bacterium, it will start degrading its own genome and die. That's the basic difference. Okay, so that's why people use CRISPR Cas9 as an editing tool for human cells, but you don't really edit bacteria this way. You can, but the efficiency is very low because most of them will die. So that's why um, some specific mutations, for example, you can only do really specifically. I want to change this A to a G. This is what we show in the paper. They need more specific tools, and that's exactly what we're applying bacteria. Just because a nuclease will kill them, while a base editor will modify the gene without killing them.
That's so fascinating. I never realized that distinction. So, did people just as a side question? Do people use base editing in human editing projects, or do they need to if they have the repair mechanism?
Yeah, you can use. It depends on what your target is and what you need to do. For some specific targets, diseases, or modifications that you need in the lab, just work with something. You might just want to have a one base pair mutation or something more or less complex insertions. For this kind of more complicated things or more specific things that you really want to have something specific inserted or changed, then you go for these base editing tools that we use in the paper, like cytosine base editors, adenine base editors, prime editors, um, transposon based technologies, because that allows you to have a deeper control on what you want to do and exactly what kind of modifications you want to insert.
So, can you use something? How do you do things like add a whole gene or delete a whole gene, like with base editing in bacteria? You just kind of mutate the stop codon or something, or do you have more leeway there?
Um, so again, this really depends on the mutation. So for example, in the paper, we introduce, so we break the start codon of a gene, but we don't really insert anything there. We just change the ATG to I forgot now. I think it's AAG. So I think we made this modification. So there's no start codon, so there's no protein being made. But that only requires the change of one base. So that's really not any insertion going on there. to do something like you were saying, inserting a gene, and that's where I'm going, like to more complex or let's say fancier base editing, or gene editing, then you rely on new technologies or technologies that are different to the base editors that we use here, like a prime editor. A prime editor will let you insert short fragments of DNA into the target site of the genome. For example, you put them together with a Cas9 or an nCas9, and it will go to the specific genome region and insert that thing there. You have transposon-based methods to allow you to insert stuff into the genome. So this technology exists. It's new. People are developing that now, and I have no doubts that they will become this kind of tools will become really really important for therapy in the coming years.
Okay, so there's a bunch of tools. Some of them play well with bacteria where they're not going to cleave the DNA and cause the whole cell to die. Some of them don't.
Yeah.
But and you guys have been focusing on this base editing to make pinpoint changes and to make it is the reason that, because I notice in your paper that it's very high effectiveness, like close to 100% effective on the population. Is that mostly, do you think, attributable to the lack of the death of the cells because of the base editing, or more than that?
Yeah, so there's more than that there, and I think we also talk about this in the paper. I think to me one of the key aspects of what we do at Eligo, let's say that there are two ways of doing this. Whenever you want to target a bacterium, you have a new bug, and then you say, okay, I'm going to use a phage to deliver something. I'm going to develop, in this case, it's a cosmid. It's a cosmid space, so it's an Eligo Biotic. It's like a cosmid. Let's talk in let's use this language for sake of clarity. Um, and you can go for really fancy phages that do a lot of things, but you don't really understand. Or you can go for a phage that you know a lot about. In this case was lambda, which is really a tame phage. You cannot really infect a lot of wild type E. coli strains with lambda. It will just not do much. Um, but the good thing about this is that you really understand what lambda does because there's so much literature on this phage. There's been so many things done with this phage that you really understand what you need to touch. The first thing we saw is like, okay, people use lambda. It works fine with wild type strains. With lab E. coli strains, but it uses a receptor that is downregulated or mutated in the gut. So the first thing we did is, okay, let's try to find a receptor. Let's try to make lambda bind something that the cells cannot lose in the gut. In this case, we modified to recognize OMP-C, for example, and also the famous tail fibers or the tail spikes that people talk about in phages. Lambda also has them, the wild type lambda list. Um, so we modify them to be very, very strong binders to the target strain. And by changing these two receptor binding domains in the phage capsid, um, we could really have binding curves that were really, really accurate. We could select variants of these eligobiotics that would really bind strongly to one thing and not to the other. We could make lambda degrade capsules of Klebsiella, of other pathogenic E. coli, just by changing and understanding what you want to target. So going back to your question, I think I started talking a lot about it. I think it's really, really important to really understand what you have in your hands so you can engineer this to make it a really a good binder. So for me, reaching that 100% was really a lot about engineering the phage particle to make a really good biotic that will really recognize a strain with high affinity, even in the crowded or complex conditions that in this case the gut that the mouse gut has.
Got it. Yeah. And does lambda, is that sufficient? Is it sufficient to only have the right receptor binding protein and then the rest of the injection of the DNA is going to happen and you don't have any more host range determinants?
So that's a good question. We have only investigated these two uh, variants from what I've shown in the paper and also other projects. This is necessary and sufficient to achieve uh, high transduction efficiencies, which is exactly what you need if you're going to have a therapy. Something that is really good at what it does. Um, I'm not saying there's something else out there. Right now, there's a lot of anti-phage mechanisms coming up. People are really taking a deep look to that. But you really have to go back to what an oligobiotic is. It's not a phage, and like a lot of the anti-phage mechanisms that you can find out there in bacteria will somehow attack the replication of the phage, or the way it assembles the once the new variants have been made, or the arrest after something starts getting transcribed. But in our case, the the capsid is a syringe, so it's the payload that is designed, and with the payload we can do a lot of things. So you can really modify to bypass all these mechanisms. There are things that exist out there. There are phages that block receptor binding uh, proteins. Like they block them so the phage doesn't see them. But since this is an engineered particle, you can go back to them and bypass this. This injection, uh, and this injection blockers too, like they block the tail tube that goes through the periplasm, for example. These features like HK97, some others that do have more genes that do block this injection. Again, you can modify these things. So I think um, it's all about going back to what I said at the beginning, and I'm repeating this: really understanding what you have in your hands, so that you can really modify it to be good or to be best at what it does. I think for me, that's really the key aspect of what we do at Eligo.
Yeah. Yeah. Uh, Back to the paper, when you guys were, trying to get it published, what was the hardest part about that process? Like, was there something that reviewers made you take an extra year to do, or what was the final nail in it that you guys had to do?
Yeah, I think required obviously, like it's a big paper. There's a lot of data in there, so gathering all this data, having all the controls, everything that we needed to show, um, it was hard. I'm not gonna lie, it was an effort, but we really really wanted to have this out, show people what we do, show people that this is possible, show people that this is a therapy that is really is something that looks like a really promising therapy, and we should really push towards that. Um, so there was nothing specifically hard about publishing process. It was the whole thing, putting all together, have in mind that we also, we're a company, so we develop drugs, so we need to also find time to sit down, get all the team members that, working on different things and say, okay, let's sit down. We need you to put this graph together. We need to dig into this experiment. Can you put this data together? And we realize maybe, we're missing this control, so we have to redo it. So it was more about finding all that time to put it together. But we were really passionate about doing this. Actually, we're really pushing hard to have this together. So I think at the end it paid out, and it paid off, and it was really, really great.
Yeah. How do you? Oh, good. Yeah. How do you think about, as a company, the value of publishing? Like, what's the goal that you hope to achieve with publishing it as a paper? Is it to attract scientists to your team, or to kind of let people know how exciting the technology is at?
Sure, I think you really touched the two most important points. So I think the first one is really to to make people aware that this exists. Again, when you talk about gene editing, people think about human genes. I think it's important that we realize that there's a paradigm shift happening right now. It's not just the human genes, it's the genes of the other things that live with you. In this case, it's a bacterium, that are really important for human health. And I think it's important that people really understand that um, that it's also possible to modify or to touch, do things to these genes, inactivate them, add things to the bacteria that cause a disease um, in the complex environment. I think that was one of the big questions when we were developing our drugs. It's like, yeah, but you guys have to use, an antibiotic mouse model, so mice that don't have any bacteria in their gut. It's like, no, we really use, you know, mice. We have to treat them with some antibiotic before so that we can put the bacterium in. But then, we run a 16S analysis and it's full of other species in there. So it's really not an easy environment. So I think that was the first thing, really make people aware that this is possible and we should all push to get there. And the second thing, as you say, like, when somebody shows excellence about what they do, can be a company, can be a lab, can be a university. I think that also really attracts talent, and that's really important. Also for us, obviously, we want to work with the best, out there, and I think it was a good way of making ourselves, like, hey guys, we're here. Look at what we're doing, and this is our vision, and this is what want to bring forward to the clinic. And if anybody's interested, please write to us.
Yeah. Yeah, that's awesome. I hope that you get an onslaught of people to come hang out with you in Paris. That's where you still are, right? Are you kind of close to the Institut Pasteur, or do you still work really closely with them?
Yes, actually, our co-founder David Picard, it's a PI at Institut Pasteur, so we still work closely with him. He has a lot of experience in phage biology. Obviously, he still work on phages. He was developing the first, CRISPR-based antimicrobials encapsulated into a phage to kill to have sequence-specific killing of bacteria. So we still work together. We collaborate with David. And physically, we're not in the same space. We started there when, 8 years ago when I joined. We had a little lab there at Pasteur, and then we moved to, I mean, you can see it's very fancy office here and in Paris. It's really, really cool. It's in Paris Center. It's a great place to work, actually. So yeah, I recommend everybody to come visit us. Yeah.
Yeah, we visited a few years back. I forget which year, but we met with Dr. Bikar, and like we talked about what you guys were up to. But it would have been like probably 2018 or something. So everything has happened since.
We moved a long way from there. So we became really good at what we do, and I think it makes me proud, to say this actually. So um, I just can't wait to really bring this forward even more and have therapy out there.
Yeah, so let's talk about the clinic and what the timelines would look like. Like, do you guys have plans to start doing clinical trials with this, or do you have a target in mind? What's the timeline for that?
Yeah, so we do have a program that is going to move to the clinic soon. It's actually this is actually a CRISPR killing. It's not a base editing. This is for treating acne vulgaris. It's actually a program to target specifically Cutibacterium acne strains that are involved in the inflammation, so the what you call acne, while leaving the non-pathogenic C. acnes strains intact. So I think that's really, really important because the therapies today uh, is the same as when you take an antibiotic. You basically kill everything, and that causes dysbiosis, and it's really not good. In the case of acne, it's really no different. So we're really excited, and we really hope that this therapy would help a lot of people that have this inflammatory disease. We do have some other projects I cannot disclose here about, but they really involve base editing in this case to inactivate some genes. There are several targets that we're thinking about, that really have human relevance in terms of human diseases. So these are really a lot of exciting things that we're thinking about. Cancer too. So there's a lot of programs that we can apply our technology to. And we go back to what I was saying before. It really highlights the importance of bacterial genes in human disease. And I think, again, that's for me, that's a take home message is these bugs have genes that do things to you that we might not have been aware of before. So I think that's the new way of thinking, let's say. Yeah.
Yeah, I want to talk about that and like how we I feel like there's been microbiome hype, and we're part in some stage of the hype cycle that's not the highest point anymore, but probably it's, still moving forward and like very solid, but we don't hear as much about, oh, the microbiome as much as I feel like we used to. But I also think there's more talk about the gut brain axis and maybe all these diseases that aren't just infectious diseases or gut diseases are related to the gut microbiota. But I just haven't heard anything recently. So I guess where are we at in terms of that, and like what's known? Like, are the cancer people saying this bug is, if only we could get rid of this one, our cancer drugs would work? And are the Alzheimer's people saying the same, or are you guys kind of connecting with those communities when you pick your targets?
Exactly. Now, that's a good question. I go back to what I was talking about before. Um, there's a lot of things happening now. There's a lot of people working on this. It's very hard to pinpoint exactly what bug is doing what. So you say, yeah, there's a gut-brain axis, for example, or there's a immune disease, and we know it must be something in the gut or in the skin or in whatever other microbiomes you can think about. But it's very, very hard to really figure out what is causing that. You see, so to me, that causation, like really, that's right, that correlation and say like correlation causation, say like this is the gene that I need to touch, and that's going to have this effect. That is what we're still trying to understand now, and most of the studies that are happening right now uh, really go for this kind of microbiome 16S analysis and say, this is the family that seems to be upregulated in these patients versus in these others, so, but we still kind of really um, attack or modify or change that these biases, if we want to talk about this in these terms. Um, so I think we need to really change the, it's not just a microbiome therapy. So what we're trying to do at Eligo is a microbiome gene therapy, which it's really different, you see. So the microbiome is still there. A lot of people, I'm sure you're going to be hearing a lot about this soon, because it's really something that is it's becoming really, really important. So, yeah.
Okay, so the hype is not burst. There's still
There's
microbiome hype.
there's still microbiome, and I think there's going to be microbiome for a while. Yeah.
Yeah, good. That's what got me into microbiology, is when I learned, I think it was in an undergrad class, that somebody said that you could transplant obese mice microbiome into the thin mouse and make that mouse obese. Like, that hooked me for life basically on microbes. And then if yeah, that was 2008 maybe.
Yeah, there's a lot of this kind of like fecal matter transplants, that happens, and it's true. It's therapies. People are doing that. Um, you can really do that. The thing is that we don't may not necessarily understand exactly how that works. We know this composition of this microbiome composition does this, and we just apply it, but we don't really know why. So it might be okay for some cases, but for most of the cases, you can't really know what other genes in this fecal matter transplant, for example, you're putting into the patient that in the future may have an effect that you don't really understand. You see, so I think it's that's the difference.
So, are you guys thinking of, as a company, like how much of the company's focus will be on kind of pushing this technology toward like using it to interrogate versus using it to treat? Like 50-50, or how do you think about that?
So for sure, we want to bring this to the clinic. Like this is, again, I think this is our vision. I think so. Eligo really wants to bring this technology forward and treat a person, a human, a patient that has a health issue, and hopefully cure this health issue. So that's the number one goal. Um, Now, as we develop our technology, this is exactly what we're talking about in the paper. We know we can apply this technology for something else that is not just applying it to a target, but to find that target. And for this, we have collaboration with some people, trying to figure out how we could exactly apply this technology to pinpoint what I was talking about, exactly what bug does what, or what gene within this bug does what. You see, so I cannot give you a percentage on what, but the most important thing is really bringing this technology to the clinic and then apply it to something else so we can keep on bringing things to the clinic. I think that's it. That's the bottom line. Yeah,
Yeah, one is upstream of the other.
exactly.
So, do you have a target in mind for what you'll go for on the base editing side? next, or are you allowed to say?
Yeah, so we do have one target. It's a really exciting target. I cannot really talk about that. I'm sorry, so I cannot really disclose. We're working hard on that. We're applying this technology to this. We're having a success. So I think it's great. It's good to see that we move for some from something that in the paper is not really. It's a proof of concept. It touches some really important genes. For example, we modify coli uh, genes, that are causing, that are known to be involved in colorectal cancer. So, it's not that we're just touching, toy genes. If we, I really don't like this kind of term, but it's not only that. We also do this with pathogenic bacteria. So we do have some exciting target for base editing going on, and I hope you will hear from us very soon. And we will let you know that we're really successful at applying this technology there.
Yeah. Oh, yes. Well, we'll be keeping tabs. I have some more questions on the science. I think our audience is probably going to be the phage directory audience for the time being, and they're mostly phage scientists. But
Just a disclosure here. I'm not a phage scientist. Okay, so I'm not a phage scientist. I'm a synthetic. Well, I'm a biochemist, structural biologist, and then synthetic biologist, and then I moved to apply all these things to phages. Okay, so I'll do my best.
perfect. I guess one thing that just stood out in the paper is the use of the PICI, or however you pronounce that. Okay, and I know we have some PICI phage scientists in the audience. And how did you come to use, like, I think there's like two different plasmids that you have to work with, and one is inducible by some agent, and then whatever product of that plasmid is inducing the next. Why do you need to use this complex two plasmid system, and how did you come to use PICI?
Yeah, so this I can answer. So that's great because it's a really an engineering question. So I'm really happy answering that. Um, so one of the I'm gonna start from the end. Okay, so the end is you're gonna treat somebody with this case is a gene therapy. Like something that's gonna modify bacteria in the mouse. Well, if somebody's a mouse, but it's gonna modify the genes in the mouse gut. And what happens after? It's a plasmid that replicates, stays there. It can be shed. So that is something that in a clinical setup may not be that desirable. So it's something that you might want to stay away from. I'm not saying it's a no-go, but it's something that if you want to, if you cannot can kind of avoid it, like you would do that. Okay. So that was the initial question why we developed this. Then um, so we needed to find a system that allowed you to have something that replicates in the production in the strain that makes the oligobiotics, but not in the strain that receives the oligobiotics. Okay, so I think for me the choice of PICI, the specific PICI, uh, was because it was shown to work with lambdoid phages. Okay, so the SAPIs, the SPICIs, the satellite phages, things that really kind of hijack the machinery of other phages. And when I saw that this specific PICI was working with lambdoids, in this case, that's great because I know the way this PICI replicates, um, lambda is going to like it because, it's kind of the natural pair. And I think for me, that was the really, really important thing. Um, Now, PICI has two basic elements for replication. It's a primase, a protein, and then it has an original replication that is a short DNA fragment right after it. So we said, okay, can we make a split system? Normally, the PICI, what it does is excises like a prophage and then replicates, and you have the primase and the original replication right after this. So it's a system in cis, so it will replicate itself. So we decided to try it in trans and see if that works. And so we have one plasmid that contains an inducible primase and a second plasmid that contains a payload that's going to be packaged into the lambda particles that contains the primase origin, the PICI origin replication, the short fragment. And that worked beautifully. So we could see that, the strains that contain both plasmids replicate nicely. There was no problem. Uh, Lambda packaged that at great titers, really compatible with Lambda, so that was great. And whenever you injected that thing into a target strain, you would get a peak of expression like a normal plasmid. I think it's about 2 hours, what we show in the paper. And then it will be lost with cell division really fast because there's no way it's not able to replicate. And importantly, these 2 hours of expression in the target strain really let the CRISPR system, or in this case, the base editor, to be expressed and do its job. So for me, it was like a really jackpot for everything, so that you could find a PICI that was compatible with this phage, that you could make it work in trans, and you could make it work even in the absence of replication in the target strain.
Okay. Yeah, I love it. So could any sort of like two plasmid system with an inducible sort of, I don't know, waterfall effect, could any sort of setup have worked, or is the picky particularly useful in some way?
I think, that's a good question. And I can't really give you like 100% answer because I don't have more examples about this PICI biology. So, and that's what I was telling you. It was, like we saw that and then we said, it looks really, really interesting. And I think we should really go for this piece, for this kind of PICI that is compatible with lambdoids. We have a lambda based oligobiotic particle. So, let's go for that. And it worked. So I cannot really tell you if using some of the PICIs would have worked or not. I suspect, some different phages package stuff in different ways, or they concatenarize in different ways. They can have like a cause, a pack, or a long-term repeats, for example. So I think that may affect the way the replication of that plasmid may affect the way the conformation that enters in the capsid. So this is more a hypothesis. Um, so I cannot really give you an answer, but I suspect, not everything will work everywhere.
Yeah. And just for the audience, what is a picky in nature? It's a phage inducible chromosomal island that I know. But what does it do, or what do we know about those?
Yeah. So PICIs, like talking about PICIs, is what I said before. So there's PICIs, there's SAPIs in some other bacteria. There's phage satellites, that also like similar to SAPIs. So what these guys do is they sense that a phage, is being induced in the bacterium. So, for example, there's a prophage that is resident. They sense that it's being induced, and then they induce themselves. So they excite themselves. They really behave like phages. There are different ways of mechanisms of action of these things. But the one we use, for example, what it does is to recognize, or know that the phage has been induced, and then block the terminase of the phage, and then modify it in a way that now the whole phage particle will package the PICI instead of the phage. So it really hijacks the phage machinery to package themselves. And then what you have, it's a natural cosmid, let's say, you see, because it contains a cos site. So they package themselves into something that looks the prophage, the phage capsid, what is there, like, that was excised from the bacteria, but inside it contains the PICI genome. So they can jump to other bacteria by doing that. See, so they really hijack the prophage.
Okay. So when you make your Eligo, what are they called? Eligo Bioscience.
Oligobiotic, yeah, it's an oligobiotic. Yeah.
Okay. Is it right to think of it like when you actually make them, you have to have a lambda phage first and a bacterial strain that has these two plasmids? And then,
Exactly. So yeah, so what you have is it's a strain. And that's actually a good point because it's also a question that I normally get. I always forget about explaining this. So we work with prophages. Okay, so we don't work with infection. Working with prophages from the engineering point of view is much better because they're stable. You can carry them in the bacteria. You have them in. You can induce them. You can modify them easily, 100%. So it's really really nice to work with prophages from my engineering perspective. Now I'm sure people in the audience will say, "Ah, yeah, but But from the engineering perspective and from what I'm working on, I think that's is the best system. So what we start with is, in this case, it's an E. coli strain that contains lambda prophage, modified or not, with the receptor binding proteins, and then two plasmids. One contains the primase that allows the replication of the payload, whatever is going to be inserted into the lambda capsid. You see, so it's a three. It's like a three element system. You see, so but there's no PICI in there. There's just the primase and the original replication of the PICI.
Okay, so the PICI doesn't have to, in your production, it doesn't have to actually sense a phage. That aspect of it is not part of the system.
There's absolutely no PICI in the strain. It's just the primase and the original replication. They are now in trans, so we separated them physically.
Yeah. Oh, so clever. Is that what genome engineer synthetic biology feels like? It's just a bunch of rigging.
Isn't that cool? Yeah, that's why I went to synthetic biology. Yeah, that's great. Like, really, we do a lot of these things. And sometimes you see, like, when you go to a synthetic biology conference, you see like these crazy systems that people build. So they're really, really cool. So,
Yeah. No, sometimes my research has like started overlapping with that kind of stuff. And it's like, oh, this is like, feels like being a kid.
yeah, it's a little bit like Lego building, you see, sometimes. It's really cool.
And you have nature to pull from, and
Yeah, exactly.
just sometimes it works.
Yeah.
Yeah. Okay, I'm just looking through my question list because I know we're getting to the end, but I kind of did some crowdsourcing of questions from some of my phage microbiome friends. But I want to make sure we hit on some of those questions to wrap up. So, one of them, so we kind of talked about the brain, gut, and the diseases that aren't infectious. And I got a sense that it's definitely of interest, and that's like definitely the future is like getting to these other diseases. So maybe we talked about that. Is it like I know you can't talk about the targets themselves, but like, are there targets that are just people are just waiting to have the technology to actually drug, like in the on the brain side? Like, is there some bug that we know that is impacting the brain, and we know which gene is the problem, and it's just like the next step is your technology?
Yeah. So exactly for the brain, I cannot really tell you. I mean, there's some kind of, you know, people and some papers say, yeah, there's neurodegenerative diseases, and that's why 1.5 curly eat the paper, but again, this is more like yeah, we know maybe something is going on, but we're not entirely sure. Um, there's we know that some bugs modify and produce serotonin precursors, and they do things to your brain in the gut, and we're just starting to understand that. Um, but I don't really think we know if exactly, this bug makes this, and that's a driver of this disease in the brain, So that I can think of. So maybe I'm wrong, and there's something out there, and but yeah.
Someone can email us and say, hey, sure, target this. Yeah. Oh, what was I going to say? I think is there an atlas somewhere, or is there a good data set that is kind of starting to catalog, all the different microbes that we know live in our body, their genes, and like matching those to disease or health states? Like, is anyone kind of curating that kind of information?
Wow, that's a huge mission. It's like it's like, imagine, like, you were it's the same as for humans, like, sometimes, you have a disease, and it's okay, go figure out what gene is doing what, you see. Um and in this case, the microbiome, different people living in different places, eating different food, having different, cultural, I don't know, like, it's climate. It's really, really crazy. So the pangenome of the microbiome, the human microbiome, is massive, is huge, Um, so I don't think that's going to be easy. I think we'll get there, but we need to have technologies like what Eligo is doing, to try to start interrogating this and say, okay, now I do have a target. I know what I need to touch, but because otherwise it's really a lot of things, and you might have, again, like families or maybe a species that tells you, yeah, maybe this bug is, this family is related to this. We've seen patients that have more of that, um, by going down to the gene, especially for bacteria, that sometimes not always. It's not just one species of species and then strains. Because if you talk about E. coli, yeah, it's E. coli, but then you have the lab strains, all the enteropathogenic strains, you have a billion different strains of E. coli. They're behave really differently, have different genomes. So, it's not that straightforward.
No. Yeah, we don't even know which bug is responsible, let alone which gene, let alone which, even to engineer, you have to be able to target, you have to know something about the receptor binding protein that would be the key to that microbe.
I think that second aspect, I would say it's easier to understand what a phage binds to. I'm talking in very general terms, okay? Than trying to figure out what the gene, what bacterial gene is causing a disease. You know, if you know that it's a bacterium causing something, or really know you're really sure, finding out this gene is not that straightforward. Uh, and and that's because the technology, again, I go back to that. We do have a lot of assays that have been developed for phages. We know how to work with them. They've been there for a long time with us, you see. So you can have more or less complex phages, but you can kind of adapt the techniques that exist to something new. But really pinpointing this gene for me in a bacterium, again in a pangenome of billions of bacteria, it's probably not that straightforward.
Yeah. Yeah. Um, and on another question that came up is related to, and this is kind of my question too, is like, can you multiplex this technology? And because I imagine it's probably not going to be one gene that's the cause. I feel like that's a minority of human diseases that have been found that are like one gene is completely responsible. How can you imagine that you would be your product would actually be, something that is going to go target like 7 different genes and just release it at once, and kind of, would it work just the same?
Exactly. So that will really depend on what you need to do. But, for example, in this case, we're using nuclease-based base editors. You can multiplex those. It depends on what nuclease you use, so we go, and it really devils in the details. But you can really multiplex that. So there would be no problem in targeting different things. Now, reaching different bacteria, you need to work around with engineering, trying to make uh, vehicles that deliver into this bacteria, but not in that one, or delivering into these two at the same time. So this is exactly, it's one of the things that we do at Eligo. So, it's like, modifying the delivery vehicles so that we have an oligobiotic that can really target different things, or with a multiplexing payload that can modify or target or kill uh, different DNA sequences. So we do all that here. Yeah.
And what about like slow growing bugs versus fast growing ones? Like, how is there a difference that you see with how effective your tech is on targeting those?
Yeah. So in principle, when you target something uh, and you deliver very well, um, the differences in maybe the bug divides slower. Like, it says it takes, I think some mycobacteria take like 2 weeks to divide something. It's like a crazy, crazy numbers. But, once a DNA payload enters in a cell and contains a promoter, does something, that response is fast. So, bacteria, if a bug takes 14 days to divide, it doesn't mean it takes a week to transcribe a gene. It makes no sense. So if you're gene-based, uh, if your payload contains a gene, will it transcribe? In this case, no place. It will be expressed within, I don't know, let's say an hour. You have a decent level of protein that will like cleave the genome, and the bug will die. So, in that sense, I don't really see an issue, a problem, or a difference. Um, it will be more about, the residence time or the payload is what we talk about in the in the paper. So, it will, if you deliver something into something that doesn't divide, if there's not no mechanism actively degrading that DNA that entered, it will stay. And then when the cell divides, it will be 50%. And then when the cell divides again, it will be 25%, you know? So that would talk about the residence time. And that's one of the things that we're trying to work, that we, kind of fixed in the paper by using the PICI. So we do have these things in mind. And again, to me, there are engineering challenges that can be bypassed. And sometimes you can find really elegant solutions like this one, So I think it's a matter of really understanding what you have in your hands, your problem, what you need to solve, and then finding a solution.
Yeah. Yeah. No, it's so exciting. Thanks so much for talking to me and just diving into like nitty gritty science and also like tell me which target in the brain we're going to be hitting.
Yeah, I wish I had an answer for you. No, thank you so much for letting me, explain, talk to you about this. It's really exciting. I'm really, really happy that I've been working at Eligo, and we keep on developing these things. I think, it's great.
Yeah. So, what do you need? What do you guys need from the phage community? Do you need people to go apply for your job openings?
Yeah, sure. We do have a job opening, so please apply, talk to us, shoot us an email if you have some target that you think, some bacterium that, oh yeah, I think this gene is important, or, I do have a phage that infects this thing, and this thing has been involved in X. Please reach out to us, and, we can start talking and maybe collaborating and find some kind of therapeutic out of that. That'd be great.
Oh, I love that. Yes. Okay. Think
Yeah.
of your gene that you say in all your grant proposals matters to whatever disease and reach out to Jesus and
Exactly.
see if he
Yeah.
wants to know what it is.
Please do. Yeah, yeah.
Awesome. Yeah. Okay. Well, excellent. And I'll also definitely point to the paper that we've been discussing, which I didn't really introduce very well, but I'll add a little bit to that. This Nature paper on in vivo base editing of the microbiome using phage-based particles, oligobiotics. They're not phages.
The phages is very important. It's one of the first things I really need to explain when I go to conference. It is not a phage. It does not replicate. It's a plasmid-based system. You see? So yeah.
Okay, well, thanks. So great to chat. And yeah, we'll be in touch soon.
Yeah. Thank you. Great. Nice to see you. Bye bye.